Is Maris. Okay. My name is Maris Röschli, and on behalf of all organizers, I like to welcome everyone to today's seminar. Before we move on to the talk, I would like to say a few words about the network more generally. So STRiVE is an ESET-funded special topic network that officially started at the end of last year, so just a couple of months ago. The aim of the network is to bring together researchers who are interested in the evolutionary role of structural genomic variation. And specifically, we want to deepen our understanding of how structural variation shapes evolutionary processes, from adaptation and genome evolution to speciation and patterns of biodiversity across the entire tree of life. We also want to build an inclusive and interdisciplinary community by connecting researchers across different fields, backgrounds, and career stages. And we would like to do this by promoting collaboration and knowledge exchange, seminars, workshops, and conferences, and shared research activities. And as a major first initiative, I would like to point out this conference that is coming very soon. So this summer, we are going to launch the Special Topic Network with a 3-day conference in Portugal. And I would like to point out that there is still a couple of open slots. Unfortunately, the abstract submission has already closed, but the registration remains open until tomorrow. So if you can make time on short notice, please register for that conference. This brings me to another initiative, which is sort of today's topic of the network, which is the online seminar series, which is organized by a subset of people involved in the network. Our goal is to host about one seminar every 1 to 2 months with varying daytimes, so this should accommodate really live attendance across different time zones. Updates and details will be shared through our webpage and the mailing list, which you can subscribe to online. Each seminar will include roughly 40 minutes of a talk by a researcher, followed by 20 minutes of discussion and questions, which you sort of post directly to the person who gave the talk. Recordings will be made available on our YouTube channel, and we will only make the talk available, so there should be no hindrance to really ask questions and go into a lively discussion. Before moving on to today's talk, I like to mention that we already have 3 upcoming speakers confirmed, so please feel free to already mark them in your calendars. So this brings me to today's seminar and speaker, Professor Loren Rieseberg. Loren completed his PhD at Washington State University. He then became an assistant professor at Claremont Graduate University before moving on to Indiana University, where he progressed from associate professor to a distinguished professor. He then moved on to the University of British Columbia in Vancouver, where since 2016, he holds one of the university's prestigious Killam professorships. Throughout his career, Loren and his research group have worked on a wide range of topics in evolutionary biology and genomics. He's especially known for his work in plant evolutionary genomics, particularly on how hybridization and gene flow contribute to adaptation and the formation of new species. Combining genomics, computational biology, and really field experiments, often using sunflowers as a study system, his research has shaped really fundamentally our understanding of speciation, invasive species, and the use of wild genetic diversity in crop improvement. Of particular interest to our network is Loren's work on structural rearrangements, and in particular chromosomal inversions. Loren was among the first to directly formulate the idea that inversions may play an important role in adaptation due to their effects on recombination. And as the title of his talk suggests, this continues to be an active area of his research. So before I hand over to Loren, a quick note about the discussion after the talk. So if you have questions to Loren, please feel to post them in the chat or the Q&A panel that you find somewhere on your Zoom. or you can also save them for later so you can ask them directly to Loren. The discussion will then be led by Katie Peikel, who tries to oversee all these questions that will come in and hopefully make a lively discussion. So with that, I would like to really thank Loren for speaking to us today and for giving the first talk in our network seminar series. And so I would say the screen is yours, Loren. So you can take
Thanks, Marius. I'm just trying to figure out how to handle these. Oh, what did I do? Okay. Does everyone see the screen? Let me know if you can't hear me or something goes wrong. Otherwise, I'll probably just sort of talk on blissfully in complete ignorance that something is wrong. So I want to thank you all for inviting me to speak to STRiVE. I was so delighted to see that this network exists. I wish we had something similar in North America since, as Marius mentioned, structural variants are dear to my heart, and I'll sort of talk a little bit about how I got there today. So I think all of you know that one of the greatest obstacles to speciation in sexual species is gene flow and recombination. That's why you're here, no doubt. And the basic idea, of course, is that divergent natural selection and drift to a lesser extent drive populations apart, and then gene flow and recombination homogenize them. And I've illustrated the population, the problem here. Here you have yellow-flowered daisies come into contact with a population of red-flowered daisies, and if you have gene flow and recombination, of course, in just a few generations, the differences are dissolved. And we've known about this problem for a long, long time. Actually, the critics of Darwin's theory of natural selection worried about the effects of genetic swamping on establishing these new traits via natural selection. And they came up with a solution, and I think all of you know this one, right? It's geographic isolation. If your populations are isolated, the problem of gene flow and recombination goes away because there is none. And this is just illustrated here again with the yellow and red flowered daisies. And many evolutionary biologists, particularly in the 20th century, really liked this idea and argued that, in fact, geographic isolation was a requirement for speciation. The problem, of course, is reality. Particularly with genomic data, we found that, in fact, speciation with gene flow is common. So it's interesting that these authors, though, sort of suggested that Darwin really didn't understand the importance of isolation. And I was influenced a bit by some of Jim Mallett's writing on this, and so I decided to go back in the literature. I was writing a review last year on, I wonder if I can get rid of this top thing because it blocks off the title. Maybe if I put it at, I don't know where I can put this. If there's a way to get rid of this.
Loren, we see your title, I think.
Oh, it's only me that couldn't see it. Okay.
Oh, yeah.
I was worried that you were missing it, and so I was thinking, well, this is not good. Okay, I got rid of it on my screen now. So I was writing this review on speciation with gene flow, and I was learning that botanists had sort of affected his views, and so I went and read some of the early literature, and and it was quite clear that Darwin actually understood the importance of isolation very, very well. This is a quote from back in 1837, where he writes, "Why are species not formed during ascent of mountain or approach of desert? It's because the crossing of species thus altered prevents the complete adaptation which would ensue," clearly with isolation. However, his views began to change, and this was mainly because of his, as you know, he just constantly writing to other people, and his correspondence was absolutely amazing. And he was corresponding with lots of other taxonomists and evolutionary biologists. I guess those didn't really exist. But he learned from botanists that, in fact, often closely related forms of plants, we'd probably call them ecotypes, sometimes species, often occurred in sympatry or parapatry, although those terms weren't used at that time. And so as he was getting close to, as he was writing The Origin of Species, he writes in his notes, no doubt here comes in the question of how far isolation is necessary. I should have thought more necessary than botanical facts seem to show it. So unlike what his critics have suggested, and this is not just folks like Ernst Mayr, but even modern evolutionists, Darwin had thought through the importance of isolation. And I think this comes out in his book as well. He says that isolation, by checking immigration and consequently competition, will give time for any new variety to be slowly improved. And this is the kicker, as he says, and this may sometimes be of importance in the production of new species. And I don't know if you've read The Origin, he sort of differentiates between animals which are more mobile, and so their isolation seems more important than in some plant groups where, of course, they're less fragile, and other groups, not just plants. And of course, we now know, mainly from genomic data, but there was data from before the genomic era, that speciation with gene flow seems actually to be common, at least in some groups, supporting Darwin's views. And of course, the question then becomes, which is the one I addressed today, and I think you all know the answer, at least one of the answers, and that is, how does this happen? And at least one of the emerging solutions clearly is structural variation, and inversions and other chromosomal rearrangements suppress recombination at heterozygotes, thereby keeping sets of linked alleles together. I've illustrated this using one of Mark Kirkpatrick's figures, which is a very simple figure. But in this case, I added a little bit. I added habitat adaptation and flowering time, sort of assortative mating rate, and a trait under divergent natural selection to an inversion, showing how this could keep these alleles together, thereby encouraging or facilitating adaptation and speciation. One thing about Mark's figure is that he shows that recombination is producing these inviable gametes because they're either duplicated or deficient. However, in many cases, I think many of us are finding that in large inversions, often there's actually no recombination at all and no loss of fertility. And so I'm going to talk a little bit about that today as well. So I wanted to give you a little history that was sort of sponsored by the idea of giving this talk. And these are some of the key papers. I've included one of mine in the key papers, which is, when you write the history, right, you get to sort of emphasize your own stuff. Sorry about that. But I thought that there was this really key paper by Trickett and Butlin in 1994, which hasn't been cited very much. It was a simple simulation, and where they just showed that the suppression of recombination between a locus under divergent selection and one causing assortative mating enhances speciation, which we could have figured out without an inversion. But it was a really important conceptual advance, and it sort of solved, the issues raised by Felsenstein about the antagonism between selection and gene flow or recombination. And then in 2001, I wrote a paper. This was based on some of the work we were doing in wild sunflowers, where we were finding that these chromosomal rearrangements were playing a disproportionate role on patterns of introgression between sunflower species. And so it was the fact that these effects were so large, much, much larger than any of the genic incompatibilities we'd found in the sunflower genome, suggested to me that recombination was the key here, not the effects of these rearrangements on sterility. And remember, at that time, everything about chromosomal speciation was about hybrid sterility, their effects on hybrid sterility. And so it was really just, there were a handful of us starting to think about the fact that maybe we were focusing on the wrong thing. And it's not that sterility, hybrid sterility isn't important. It's simply that it's much less important than recombination. And at the same year, Mohammed Noor wrote this paper I thought that was quite influential, basically showing that inversions can maintain Dobzhansky-Muller incompatibilities in the presence of gene flow. And so this was, I thought, a really important advance. I remember, though, when I had submitted this paper to the Speciation special issue in STRiVE, and one of the reviewers, I think it was Mark Kirkpatrick, but might have been Nick Barton, they asked, well, how do these rearrangements become established? And I didn't know. I did some hand waving. I talked about, the one of Brian Charlesworth's papers on, how co-adaptive gene complexes could help to establish inversions. So it's essentially an epistatic model. But I didn't know the answer. And so it was really exciting in 2006 for me, and I think for a lot of people, when this paper by Mark Kirkpatrick and Nick Barton came out showing that suppression of recombination between locally adapted alleles favors inversion establishment. And so what I'm going to do today is, this is sort of the organization of the talk. What I want to talk a little about is some of our early observations that got me thinking about the importance of recombination suppression. And then I'll go through a case in sunflowers where we linked these inversions, in this case, to local adaptation and reproductive isolation. And then I want to just talk a little bit about some of our studies that we've been doing recently about how large inversions become established, because I don't think that the Kirkpatrick and Barton model has been fully demonstrated, and also it's not, I think, quite sufficient on its own. And I'm going to talk about the underappreciated role of introgression in the process. And then I'm going to talk a little bit about how sunflowers avoid the maladaptive effects of recombination suppression. And then finally, I'll talk about some new work on the distribution of inversions across the eukaryotic tree of life. So to introduce you to sunflowers, there are about 50 species across North America. What's important about them for this talk is that the populations are tightly adapted to local environments. And so in sunflowers, and I think most plants, local adaptation is the first step in speciation. And so, we started working on this before the word genomics, I think, had come up. We started making these genetic maps back in the late 80s and early 90s. And this is a case where we're looking at certain relationships among three wild sunflower species. I think these were certainly the first maps of wild plant species. And you can see we're working with just very low resolution things. And so we were able to pick up some translocations and a handful of inversions, but we missed most of the inversions. We just didn't have sufficient resolution. And one of the things that we also did was we began using these maps to look at patterns of introgression, both in experimental backcrosses and in natural hybrid zones. And the big thing we found, which is sort of illustrated, so this is one of the species pairs we've worked on a lot, Helianthus annuus, that's a common sunflower, the prairie sunflower, and then there's a hybrid between them. We just caught this car in the background. So we took the photo. Anyways, what we found was in general that there was much less introgression across the rearranged regions. Also, they tended to act kind of like a chunk, a big chunk, whereas we got much more heterogeneous introgression across collinear regions. These papers were mainly published in the last century, but we kind of repeated this work with whole genome, with transcriptome data and whole genome sequence data over the next 10 to 20 years. And of course, we got pretty much the same results with much better resolution. The one thing I wanted to mention, I brought this up, and so it was these kinds of data that really made me write that paper in that review in STRiVE. But one thing I wanted to mention about this is that we really were looking at the wrong thing. And we were looking at differences between species, between really what we call good species. And it turned out all of the action, or most of the action actually is within species. And I'm going to give you a case study sort of illustrating this in a case where we're looking at two ecotypes. We've done similar kind of case studies in other systems in wild sunflowers, but I think this is the best system. And this is between dune and non-dune ecotypes of Helianthus pedularis. And it was actually one of my grad students who's now a prof at University of Colorado, Nolan Kane, who discovered the dune ecotype of Helianthus pedularis. We're probably going to name it as a different species since it's really quite distinctive. And so it occurs on this in the Great Sand Dune National Park and Preserve. And the big difference between them is the big seeds. There's these huge seeds on the dunes, and then on the non-dune ecotype, which is just normal Helianthus pedularis, they have much smaller seeds. And this gives you sort of what it looks like. These are megadunes, the largest dunes in North America. And then you have the sand sheet below. And then behind the dunes, you have the forest. And we found that this sort of system is repeated multiple times in North America, once in Texas and once in Saskatchewan, but it might be found elsewhere as well. There are smaller dunes, but they tend to have many of the same inversions involved. And in addition to the seed size differences, the dune plants flower later, have greater nitrogen use efficiency. So the dunes are very low in nutrients. They aren't low in water, though. The plant just has to sink a taproot into the dune, and they're not water limited at all, whereas there are water limitations off the dune in the sand sheet below. And so we began studying this, in I think around 2007 or 8, and much of the early work was done by Rose Andrew, a former postdoc who's now at the University of New England in Australia. And one of the things she did, this was using RADseq data, she estimated both the divergence time of the dune, non-dune ecotypes, which is about 5,000 years ago, certainly less than 10,000 years ago. And she also estimated levels of gene migration between the dune and non-dune ecotypes. And what she found was the gene flow was moderate, but high enough to prevent differentiation through drift. And so, but really not high enough to completely stop divergence through natural selection. And so we struggled working on these dunes, and for reasons that you might imagine, most of our experiments get destroyed. A problem we have until today, and there are several reasons for this, but the biggest one is quite obvious. Dunes move, and so you get a big windstorm and your experiment's gone. But I had and the other problem is that they're pretty much the only green things. Well, there are a few other green things, but and so if you transplant in seedlings like what we used to do, they just typically get eaten by something by the next morning. And so, but this is Kate Ostovic. She's an assistant prof at University of California, Riverside, and a remarkable biologist. She managed to make this work. So here you're seeing a reciprocal transplant experiment between the dune and non-dune populations. And Kate did one of these really comprehensive studies of the reproductive barriers across the life cycle of the dune and non-dune ecotypes. And I've summarized it here. It's some ways better to look at reproductive isolation of the dune ecotype and of the non-dune ecotype separately, but I've summarized them here to make it a little bit easier. But what you see is that the strongest barrier really is immigrant inviability. And this is mainly because the non-dune ecotype seeds, those small seeds, do not survive in the sand dune. I don't think we've ever seen one come up. Whereas the dune seeds will survive on the sand sheet, but the dune plants produce so many fewer seeds that it is a big barrier. And then flowering time, it's a small one. There is a flowering time difference, but they overlap so much that it doesn't contribute that much to reproductive isolation. A much bigger barrier is pollinator isolation, and this is mainly because the dune ecotype is found on the dunes, which has a very different set of pollinators than found on the sand sheet below. And Kate actually found two new bees to science. And then we also found, this was a surprising thing to us because these are quite recently evolved, but there's a difference in conspecific pollen precedence. Basically, we don't know how this happens. Kate is trying to figure it out, but dune moms prefer pollen from dune dads, and non-dune moms prefer pollen from non-dune dads. And we don't know how they do it. It's something we're trying to figure out. There was no difference, though, in hybrid viability or fertility. We did see selection against hybrids when we transplanted F1s and back crosses into the two different environments. But the total isolation is really quite strong, 0.99 to 3 to 0.996. And so this still does allow a trickle of gene flow through if you have large effective population sizes. So it's not, it actually is consistent with the migration rate data developed. But you can see it's strong enough that we think they probably are good species. So where do the inversions or other structural variants come in? Well, we did large GWAS and GEA type studies. So you can think about this as environmental GWAS or GEA, as well as just sort of looking at phenotypic data. And this is mainly done by Marco Tedesco, who's now a assistant professor here at UBC. And what you can see is you see these large sort of non-recombining blocks that underlie the main dune-specific traits like larger seeds, flowering time, and this is cation exchange capacity. It's a measure of soil fertility. And so this is a GA analysis done with, can't remember what program. But it basically, these are the inversions also involved in adaptation to low nutrient soils. And if we compare this to an FST analysis, what you see is that the almost all of the differences between the dune and non-dune ecotypes actually map to these haploblocks, which turn out, as you I'm sure all know, are inversions. And we've sequenced lots and lots of different genomes. Sunflower genomes are big and they're hard to sequence, but we now have, and we're now doing really detailed comparative analyses across the genus. But the number of inversions in sunflowers is just crazy. So this is just showing these, when you get this is between Helianthus pedularis and the cultivated sunflower, but I could have shown you one of the two different haplotypes within pedularis. But what we're seeing, I just thought I would just show you by the way, this inversion on chromosome 14 is one of the three that was in that last one of those three haploblocks. But there's about, 60 to 70 relatively large inversions between Annuus and pedularis, which are relatively closely related species that still can hybridize. And then these things are these translocations. And it's the translocations that mainly cause the loss of fertility in interspecific crosses. The inversions don't seem to have much of an effect at all. Okay, so one question that might come up is, well, how did speciation occur so quickly? And we think what happened is these inversions or haploblocks likely were recruited from standing variation. And keep in mind that 10,000 years ago, during the last glacial cycle, there were no sunflowers on the dunes. It was too cold for them. And so presumably these inversions were retained in the standing variation in the sort of non-dune ecotype, or the sort of conventional Helianthus pedularis, and then were recruited when the dunes were recolonized. It's quite similar, I think, to the stickleback story, where over and over again the inversions have been used to colonize freshwater habitats. And so we think this has just happened again and again and again, and each time the divergence gets a little bit greater. So now I want to sort of switch gears and talk a little bit about how large inversions become established. These are sort of some ideas I've been sort of thinking about over the last few years. And this illustrates the Kirkpatrick and Barton model, right? You have what you need is you need alleles that are locally adapted to one environment. And then the alternative allele or different set of linked alleles are adapted to the other environment. The problem, of course, is getting through this adaptive valley, right? Through the heterozygous stage, particularly if the heterozygotes are underdominant or show some level of loss of fertility. And so I've kind of illustrated this. The big problem is going through this adaptive valley, right? And so there are a few things and so an inversion can be favored, right, if it can hold these locally adapted alleles together. The big A, big B, and big C in this habitat, the little a, little b, and little c in this habitat. This is supposed to be an inversion, by the way. And so there are a few expectations, right? Inversion heterozygotes must be selected against. We need to have sympatry or parapatry between ecotypes or species. Otherwise, this model doesn't work, right? Or at least between different locally adapted, different environments. And then one thing that I think hasn't been discussed very well is that the underdominance of the inversion heterozygotes can't be very large at all. If you look at the model, it takes really quite, it only works if you have very weak underdominance. Otherwise, it becomes, just unlikely. And so I think this observation that we've been finding is that most large inversions don't show a loss of fertility is telling us something. It probably has to be. Otherwise, they wouldn't have become established in the first place. So we've done some of the kinds of studies needed to do this. This was a student of, this was Kate plus April Giebel, a student of Nolan Keynes. And when they planted out all those backcross plants, they also were able to measure the strength of selection against inversion heterozygotes. And not surprisingly, they are actually strongly negatively selected as predicted. There's an exception here, which this is for chromosome 14, by the way. I don't know why. Something we need to replicate. I have a student now working on sort of the effects of geography on inversion establishment, Yua Yu. She's senior PhD student, should finish in the next year or so. And she's been working on a complex of sunflowers in Texas and Florida. It's the Helianthus debilis praecox complex. And these two regions differ greatly in terms of sort of the extent of habitat heterogeneity um, and coexistence of different forms. And in Texas, this is where the annual sunflowers, at least, and maybe the entire genus originated. And there are many, many species. There are 11 species or subspecies of the annual clade, all in Texas. Most are sympatric or parapatric, all hybridized with at least one other taxon. In Florida, there are 4 species or subspecies. They are mostly allopatric. There's one case of parapatry, and no hybridization has been reported. And so I thought that they kind of illustrated the difference. And of course, this is a sample size of 1, right? So take it with a grain of salt. But I thought I'd show a little bit of Yua's sort of recent data. So what she's done is she's done some reference genomes, and we've been finding that just comparing the 2 haplotypes of individuals turns out to be a really nice way of in haplotype result assemblies turns out to be a real nice way of sort of looking at structural variants. And I'll talk more about that with this larger, broader comparative study. But what you see here, so these are 2 haplotypes we only have 1 genome for the sympatric subspecies, but you can see there are lots of inversions. This is mini-map of the 2 haplotypes, and all of the orange areas are these, lots and lots of inversions. This is the allopatric subspecies. We only found 2 really decent sized inversions. There are a couple smaller ones, but the difference is quite remarkable. And this is between subspecies, so you can just get an idea of just how many inversions there are. And we're trying to put these on trees to sort of figure out exactly what lineages they arrived in. But it illustrates the importance of sympatry and parapatry. And she's done this she's gotten the same results with using population genomic approaches to find segregating inversions or other haploblocks. So I do want to, so this is, so the other thing I think that's important is the observation that many large inversions that are involved in local adaptation, actually, they're highly divergent, and yet they have no effect or very little effect on fertility or viability. And so, and this just shows in sunflowers, this is from a paper that Marco Tedesco led, but Greg Owens and others were really involved in this a lot. And what you see is that all of these haploblocks, it turns out that we now have data that I think 35 of 37 turn out to be inversions. And even those that aren't inversions at least have at least one inversion involved. But anyways, what you can see here is that they tend to, the divergence between them is quite, they're all old except for this one. And when we do look at them phylogenetically, it looks like that they all are derived, pretty much all are derived from introgression. This is something that's in progress, so I don't have a final count on this. And this is just an example from Helianthus argyphilus, the silverleaf sunflower. We have this 30 megabase haploblock on chromosome 6 that explains most variation in flowering time between two different ecotypes of Helianthus argyphilus. And by the way, it's due to both a deletion, a series of deletions and an inversion. And when we look at this phylogenetically, you can see that the early flowering haplotype of Helianthus argyphilus, this is the one with the functional gene of flowering locus T. So it actually flowers early, and it is a very recent derivative of Helianthus annuus. It probably was derived in the last 5 to 50,000 years. We don't know the exact date, but certainly hasn't been there very long. And then it's quite divergent from the late flowering haplotype, which clusters with sort of where Argyphilus should cluster. And so they're about 1.8 million years divergence. But you can see that, in fact, the inversion arose very, well, not inversion, the haploblock arose very, very recently. And so we think that this is, and so why does this matter? And we think that the divergence of many inversions might be due to introgression rather than ancient origins. For example, and the divergence we suspect, and we don't know this yet, contributes to asynapsis and a complete absence of crossovers. So there's no inversion lupus formed, no loss of heterozygosity in heterozygotes. So, and so we think that this is why introgression is really key to the establishment of large inversions. It just reduces underdominance. And there was a paper by Jeff Fader in 2011 where he was trying to discuss why inversions in Ragolitis had originated through introgression. He called them originating in allopatry, but basically they were through introgression from another species. And he pointed out that geographic isolation prior to introgression also facilitates the assembly of locally adapted alleles. And I think that's correct as well. Although my suspicion is that his model is fairly convoluted. You have these inversions arising and being at very low frequency in allopatric populations, and then somehow come into contact with the other species. I think what's probably happening is that many of these inversions actually arise directly in contact zones and then are driven to high frequency. And the ones that are driven to high frequency are those that are highly divergent and don't cause a loss of fertility. It looks like I'm running out of time, so I'm not going to talk about the recombination suppression. I'm going to go directly to the talking about the comparative analysis, which I think you might be more interested in. So just to let you know is that it turns out that the reason that so sunflowers show very little accumulation, inversions show very little accumulation of deleterious mutations, and it's mainly because they tend to be homozygous a lot. And so the idea here is that in sunflowers, environments are heterogeneous, right? And so the idea is that inversions tend to be found spatially segregated often. And so they tend to be homozygous much more often than they are heterozygous. And so this just shows the homozygosity of inversions is higher than sort of your average SNP. And this is why we don't seem to see the accumulation of deleterious alleles in any greater fraction than over the rest of the genome. Okay, I want to go here because we're sort of running out of time, I think. And so this is some recent work done by one of my postdocs, Xu Zhang. There were other people involved, but he was the main driver of this. And what he's done, and I know some of you are working with, I know Claire and others are working with Darwin's Tree of Life data set. And so what we did was well, what Xu did really was he found about 1,100 data sets or for individuals. Each of these is for a different species. We typically only use one species, one genome per species, and we tried to have a fairly equal distribution across groups as well. So, but anyways, many of these come from the Darwin's Tree of Life project, and then for plants in particular, we took data sets that were publicly available. And what Xu did was he reassembled all of these, creating haplotype resolved assemblies, and then just looked at inversions between the haplotypes within each genome. And this allows a fairly unbiased way of looking at sort of the distribution of inversions before they've been filtered out by selection when you compare them between species. And just to highlight a couple things, one is the important thing, and I think all of you know this, right? You wouldn't be in this STRiVE network otherwise, but inversions are common across eukaryotes. One of the big surprise is that they're actually more common in animals and fungi than plants, and this was a big surprise to us. I think on average, the median was about 90 some inversions in animals and about 45 in plants. But the other thing which we found, which was interesting, is that large inversions are much more common in plants. And these are the kinds that really are involved in local adaptation. And you can sort of see this here. So more of the genome in a plant is covered by inversions, but there are more total inversions in animals. The other thing is that the inversions in plants tend to be much more highly divergent. And so here we're using pi and the inversion over, that is expected heterozygosity or gene diversity and the inversion over the rest of the genome compared to the rest of the genome. And it's also interesting that as inversions get longer in plants, pi actually, or the divergence of the inversion goes up, whereas in animals it goes down. So large plant inversions tend to be highly divergent. Large animal inversions do not. Of course, when we're doing this, there are going to be exceptions for every, different animal groups are going to be very different. I imagine that animal groups with very low vigility, like sea snails, will probably act a lot like plants. But we haven't been able to do that yet. It's interesting also that the plant inversions have fewer genes and more TEs. And this gets this TE effect and it's interesting that in animals, in fact, the TE content goes down with larger inversions, and in plants it actually goes up. And I think this is important as well because these TE regions that are going to diverge most rapidly, right? And so I think this really helps in plants the establishment of large inversions and probably leads to greater asynapsis of large plant inversions when they're established through introgression. All right, so I'm gonna, so that's all very speculative, that last bit. But I want to sort of summarize. Obviously, sunflower ecotypes and species are differentiated by numerous large inversions, crazy numbers between different species, but apparently there are even more in animals. So um, they contribute importantly to local adaptation and reproductive isolation, and they're caused by large inversions and other structural variants, which act as recombination suppressors. I think this is repeating things that all of you know. They're ideal recombination modifiers, though, and I think this is why they're so important, is because recombination is suppressed in heterozygotes only. So you actually don't have as much of a problem with accumulation of genetic load as you would with other recombination suppressors. And they play a major role in evolution by, of course, reducing recombination between locally adapted alleles, and in some cases, those contributing to reproductive isolation, as in the Kirkpatrick and Barton model. They are abundant in other groups of plants, animals, and fungi. Interestingly, they're larger and more divergent in plants, perhaps suggestive of a greater role in plant adaptation and speciation, similar to sort of maybe providing a mechanism for Darwin's hypothesis about the greater importance of the less of a requirement for isolation in plant speciation. And obviously, I think this offers a general solution for how speciation can occur in the presence of gene flow and recombination. And I want to end by acknowledging the folks involved. The Darwin's Tree of Life data is just an incredible resource. And so I want to thank everyone involved in that. And then these are the people, I think I've mentioned them all during the talk. And my funders were mainly been from NSERC, but I also have some funding from Genome Canada and Genome BC. And so thank you all very much.
Thank you, Loren, for this great talk. We don't have a little clapping thing on the
Oh, that's fine.
So I think there are definitely time for questions, so people can either raise their electronic hand or put questions in the Q&A panel that you can find at the bottom. Give people a moment. I'm just not seeing. Just give everyone a second. Claire has a question. Go for it, Claire.
Maybe I can get started while people are start
Yep.
to figure out the Q&A. I'm curious about the pattern you see with the TE going down in long inversion and up in plants. Have you seen if the TE are localized at the breakpoints or all along the inversion themselves?
So so in plants, I think part of the reason we see this explanation is plants have these very large pericentric regions. And so if you have a large inversion, there's essentially no way you're going to avoid them. And so you end up, and so any large inversion, you end up getting into these pericentric regions. And there is a downside of this.